Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on November 13th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 67th episode of Matters Microbial. Thank you all for listening and or watching and for continuing to spread the good microbial word about the podcast. It means a lot to me. I was delighted to receive an email from a listener about last week's podcast with Dr. Pat Schloss about studying the microbiome. Rona wrote the following. Dr. Schloss was talking about how he would ideally like to know how his fecal microbes change from year to year to see if it's an indication of something, particularly if there is a malady associated with that batch of microbes. He spoke of how he would love for his annual physical to include this information to see if there is a trend that could be associated with a potential malady. It made me think, is a once-a-year fecal sample indicative of only how we are on that particular day? Do we know how our microbes change week to week anyway? Is a once-a-year sample indicative of what we represent? Do our fecal microbes change week to week or season to season? Has there been work done in which one logs their fecal microbes over time? Do seasons have an impact on one's microbes or one's own weight loss and diet? So I asked Dr. Schloss, who replied as follows. That's great that Rona asked a question. I would say it's a matter of magnitude of change. There are day-to-day changes, but we experience these under a condition of health. What I would be interested in seeing are the changes that are larger than the day-to-day variation. My hypothesis would be that the larger changes are associated with a change in health or some radical and constant change in diet, that kind of thing. By analogy, we might think about body temperature. Your body's temperature changes over the course of the day and certainly has day-to-day variation. But the big changes in temperature are the things about which we worry most. So I was very happy to share Rona's question, thank you, Rona, and Dr. Schloss's answer, thank you, Pat. Things are proceeding well in my Bio 350 microbiology course here at the University of Puget Sound. We're getting closer toward the end. I myself collected some seawater samples from Puget Sound and enjoyed a few sneaky, tiny glowing colonies in the sample, as you can see here. I wonder what their role could be in that environment. Regardless, they're lovely, like tiny gemstones in the night. Micronauts also showed me some fascinating colonies on some of their plates, either from seawater samples or lichen and moss samples. As you can see with both photos, there is a strange wrinkly surface to the colony. It reminded me very much of Dr. Roberto Coulter and co-workers' research showing that wild bacterial colonies can look very different from their domesticated lab rat versions. I'll put a link to that article in the show notes. It's really worth your time. My micronauts have isolated some bacteria from their lichen and moss samples and are currently testing them for the ability to produce antimicrobial compounds, as you can see in this image. Successful isolates lead to a zone of inhibition on a background of the test microbes. This is actually part of Joe Handelsman's and colleagues' crowdsourcing antibiotic discovery program for students, Tiny Earth. Again, I'll put a link to this wonderful program in the show notes. Moving on to today's topic, I have always been interested in symbioses, that is to say, organisms that live together. In middle school, I was fascinated by Chlorhydra verdissima, seen here, a small selenorate that has a symbiosis with chlorella algae. How did that association occur, and what types of chemical communication is taking place between the partners? During my PhD work, I became interested in Cynorhizobium melilodi and its ability to form nitrogen-fixing root nodules on alfalfa. Here's a nodule you can see. Again, the signals back and forth between the bacteria and the plant are intricate and fascinating. Even my love of bioluminescence took me to the beautiful bobtail squid and its luminous partnership with Vibrio fischeri. Once more, the chemical language between the partners is wonderful, complex, and a puzzle to be solved. So many questions. Thus, it's a real pleasure to welcome Marcy Balunas of the University of Michigan to the podcast to discuss the work her research group does exploring the chemical languages used by several symbioses. Welcome to the Quality Quorum today, Marcy.
Thanks for having me, Mark.
That was a lot, wasn't it?
It was great. It's nice to hear everyone's take on symbiosis.
Yeah, well, I enjoy it very much. And I think the first thing for listeners and viewers, and I often talk about the fact that we have what I call beginning micronauts and advanced micronauts. We have both here. And that's my goal with this podcast. The definition of symbiosis becomes important because people have strong feelings about it. Would you care to give your definition?
I can. Just keep in mind that although I am now a microbiologist, my training comes from chemistry. And so when I have delved into the sort of intricacies of symbiosis and of microbial, not microbial language as we'll talk about it with chemical communication, but instead the way microbiologists talk about organisms, I had to learn the difference between commensal and beneficial and symbiont and pathogen. And now there are all sorts of other words like holobiont, and there are others that I don't even know that I could remember at the moment. And so for me, I am most interested in the types of symbiosis, as you mentioned, I would think of as the organisms living together. But I actually am much more interested in the ones that are beneficial. And so bacteria, I suppose we could think of other microorganisms, but I really have focused most of my independent career on bacterial chemical communication from beneficial microbes. And so the squid is the perfect example of where to get started thinking about this, in the sense that the Hawaiian bobtail squid has the light organ that has a Vibrio fischeri in it that bioluminesces. And so lots of people know about this organ. What they don't know as much about is that female squid also have what's called an accessory nidamental gland, or ANG. And that's another organ that has, instead of one bacteria, it has many bacteria in it. And so and those bacteria, my colleague Spencer Nyholm and I have shown over the course of our research together, those bacteria get put into the squid egg from the female squid, and they are protecting the eggs while they are after they hatch, before they hatch. And so they protect the squid eggs before they hatch, so that they do not become overgrown by fungi or other bacteria or predators, etc.
May I ask a quick question? So is this mostly antifungal, do you think?
So that's a good question, Mark. I would say yes, that one of the best ways to think of this, the ants that we study, honeybees that we like, there are a lot of these systems where these beneficial bacteria are in some way, and again, we could argue, all the microbiologists I know would argue about using the term beneficial. I think a lot of them feel much more comfortable with commensal. But I think in the case of the ant and the squid, we know they're beneficial. So I feel like I'm comfortable with it. Whether or not other microbiologists are is a different question. But I do think they're mostly antifungal. We have work that shows that they're also antibacterial. And we've been looking at their ability to modulate the immune system as well, because we think that the host, the eukaryotic host, whether it be the squid or humans, so we work now on human systems and murine mouse systems, that the host has some initial reaction to a lot of these bacteria. And so the bacteria that want to stick around are likely, the hypothesis here is that they are likely to change the immune system in hopefully not a terrible way, because we don't want the systems to be super depressed, but even still, that they're changing the immune system. So I think that they have a sort of a bunch of different possible functions. The other that I didn't, you asked about fungi, but I still think that there's some, especially with the squid eggs, there are predators that would come and love to eat those eggs. And so I think that they likely include cytotoxic or other types of molecules. And our thought is that those cytotoxic type molecules have to not kill the host. They can't kill the squid egg, but the idea would be that potentially they are warding off other predators. So we'll see.
I'm gonna like backtrack a little bit, and just for listeners and viewers, I want to just emphasize a couple of things. So the first thing is a good definition of symbiosis is from bad Latin, as I was often told, to eat at the same table. And when you think about an association that way, you know, we're kind of used to the macro level. And what I tell people when I discuss symbiosis is that I'm going to call it a ballet, because it is, between a microbe and its macroscopic partner becomes very, very important. And it haunts my dreams because it's very important. And you have several different systems that you're going to talk about. And I want to just emphasize that we tend to look at things according to our biases. And a good example is the old saying that to a carpenter, every problem is solved with a hammer and nails. To a bioinformaticist, everything is solved with a genome sequence. To a geneticist with my own training, a transposon insertion to a gene into a gene, a knockout is everything. None of that is really true. We're in an age where we can look at large numbers of associations, and we've talked about this on the podcast several times, and that brings in different points of view, different types of training. I'm wearing a pin that says jargon excludes people, because it often does. That jargon may be necessary between people who are experts in a field, but at the same time, I really want to make clear to everybody this is a fascinating thing to be discussing. And the other thing that I want to make sure that I emphasize is that you're trained as a natural products chemist. And so here's something that biologists don't think about. So for example, in the image that I showed a few moments ago, where there's a bacterium that I put onto a background of plate and there's a zone of clearing, what chemical is being made? And people like me don't know. And so then we try and do really simplistic things. People like you have real power to determine what those chemicals are. And I think it's clearly been very productive for you, looking at the diversity of interesting systems you've been looking at.
Absolutely. And I think that thinking about plates in general, there is an image of a plate that we can share with you about more than one microbe sort of competing with each other. So the very sort of bringing you from a traditional microbiology background and perspective to then thinking about the chemistry. And so on that plate, there's a highlighted section that's very sort of colorful, and that's a molecule. It's one particular molecule. In this case, we actually don't know what that is, but these are 3 gut microbes from the fruit fly, and they produce different things in different zones, and we're excited about figuring out what that is. And so we used what's called multi-imaging mass spectrometry to get that image, and that image shows that molecule in that one spot, right where those things are touching. And so, again, so I think of much of the chemistry that we look at the beginning and as really driven by the biology. And so, for example, there's another slide that in which we can talk through sort of our idea of interaction driven molecule discovery. And so, how do we decide what molecules are important? And the answer to that question is by using the zone of inhibition assay, for example, or by doing a huge suite of assays in different ways, in different strains, different types of things, and prioritizing using that biology. Or in that particular slide, it talks about the ecological insights. And so, as we got started thinking about why these eggs, squid eggs in particular, because I think that's what's shown on the slide, we got started thinking about, well, what's going on? Why aren't they covered in fungi all the time? And the reality is that the answer became this huge research project and a collaboration that's now spans almost 12 years and hopefully the rest of my career, looking at the Hawaiian bobtail squid because it's so amazing. And the chemistry and what I bring to the table in that collaboration isn't the 20 years of knowledge about the squid and the bacterial associates. It is instead the idea that the bacteria and the host, in this case the squid, are talking to each other,
Mm-hmm.
and or using that chemistry to protect against this fungal fouling. And so, yeah, so that's what we do. And I agree about jargon. Being a chemist coming into microbiology, the jargon was a stumbling block at first, for sure.
And well, many people misunderstand me. It's very much, you need a vocabulary in science. If you were to learn a foreign language, there's a vocabulary that you need. And I certainly agree that there's a vocabulary that you need in a lot of forms of science. But one of the things that I have my students do, and it's very important to me, is write a version of their term paper, one page, that's meant to be for an English major, not a STEM major. And they find it very challenging. And then they get into it and they understand that that's how you really get what you're talking about. So this idea of this interplay is very interesting to me because when I was in graduate school and I'm looking at the Rhizobium root nodule symbiosis, both sides are definitely talking to one another. And we have some idea of what those signals are. That's why I call it something like a ballet. So the squid system that you looked at is a very interesting one because, as you mentioned, most people have focused on the bioluminescence aspect. The one microbial god knows how much I care about that, and I do. But also this idea of the accessory gland protecting the eggs is a fascinating one, and it has relevance to all kinds of things. So here you have a population of bacteria, and it may not be, I don't want to steal your thunder, it may not be one type, but interplay between those organisms that create compounds that act as a helper to protect those eggs. And again, this is with the Hawaiian bobtail squid, is that correct?
Yes.
Because there are lots of other squids that have these nidamental glands, and I'm really happy you're looking at the bobtail squid because we know so much more about it.
I think that's probably why we started. Although, to be honest, one of the things that we're doing that we've sort of moved that project into is thinking about climate change.
Yes.
And so, and the effect on the eggs. And the reason this relates to what we're talking about is that we think that the eggs they're made of jelly, and so they have an embryo. I don't have a picture of this, I'm sorry, but they have an embryo on the inside, and they have an embryo, they have jelly coats around them, and the bacteria are in that jelly coat. And a squid egg is about the size of my pinky nail. And the whole clutch is probably smaller than about the size of a golf ball, maybe a little larger. Golf ball, tennis ball. Think about what somewhere in that size range for the whole clutch, and those get laid on the seafloor. And you can imagine that if temperature changes, that the squid has to change what it's doing, and where it's laying its eggs, which things it's subjected to as either a predator or for biofouling. All of these kinds of things play a role. And so we got started looking at the Hawaiian bobtail squid because my collaborator has been working on them for his whole career. But we have started also, in fact, we just got started, just submitted a paper earlier this week on the tunicate. And so there's a nice picture of our study organisms. This is not all we're limited to, but in that image, there's in the upper left-hand side is Bacillus schlosseri, which is pretty funny because it's almost the same last name as your previous speaker. But Bacillus is a marine tunicate. It's a colonial tunicate that is a pretty amazing system all by itself. Tunicates are one of our closest living invertebrate relatives. And so they have loosely defined organ systems, but they have a branchial sac, they have a stomach and a gut, they have gonads, and then they have excretion pathways. And so they can be solitary and quite large. And so the ones we've collected these in Panama, as well as Alaska, as well as Connecticut, which is where this image is from. The largest tunicates I've ever seen were at both extremes. And so the Alaskan tunicates that we have, I have an image, but unfortunately it is on my wall outside, not in my wall here. But the tunicate was probably the size, I would say about the size of a hand. And that was the same in Panama as well. But interestingly, we got really excited about this Bacillus species of tunicate because it's colonial, it's invasive, it has lots of interesting sort of reasons to be excited in the macroorganism. But many tunicates are known to harbor it's less known if they are commensal beneficial bacteria, but they definitely have a suite of symbiotic bacteria. So they could be transient, they could be part of the system. And so we have used chemistry and genetics and now metals, actually, to study these particular tunicates that we're interested in, to look at the interplay between the microbes that are producing these molecules within the host, within the Bacillus tunicate, and what they're doing with metals, because we are starting to get more and more interested about metals and how they interact with host microbe symbioses.
I think that's really interesting. And I also know that many natural product chemists have looked at tunicates and looked at the microbes associated with them and found some of the compounds being made are bioactive and in some cases potentially of human use.
Yes, there's a cancer drug on the market now from a tunicate.
Yeah.
And there's some conversations and some really recent research on that being potentially made from a microbe that lives with that tunicate. Mm-hmm. I don't think that is a clear defined story yet, but it's getting there.
But it is interesting that we tend to be super reductionist. And we look at I've said it before, single colonies on a plate, bacteria growing in a culture tube. Those things don't occur in nature. And so I think a real strength of your approach is you can look at the interactions between these organisms in a way that I think would be very hard to do in a culture-dependent fashion. Yes, you can come up with ways to do it. Don't misunderstand me. But I think that you can find things that people haven't seen before.
Yes, we have a lot of fun finding new compounds. It is the hardest part of what we do, I think, because just like I told you about that plate in which the MALDI imaging mass spectrometry that we had done, we saw this molecule only at the interface of the two bacteria, we don't know what it is. And so really there's a whole part of what we do that is to define the unknown.
Yes.
And so there's what's called targeted mass spectrometry and untargeted. And we do we do more of the untargeted version. And so there's a sort of pipeline of what we do to figure out what things are. And so we start with this idea that we collect a whole bunch of mass spectrometry data. And so I mentioned MALDI. That's a new field for us, although many other scientists work on MALDI all the time. We instead work on what's called LC-MS-MS, or liquid chromatography mass spectrometry, tandem mass spec. It depends on how you want to call it. And that's the first step. And so after we extract things, we put them into the mass spec, those extracts into the mass spectrometer. And then we have to figure out what to do with these massive suites of data. And I like to call them haystacks because they really are haystacks. They are these just a bunch of dots on a screen, really. And so one of the other things that we can look at is the how we prioritize. And so we can prioritize based on the experimental design, which is what we do most of, is taking these experiments, making sure that we design the experiment both in the field and in the lab. And you're right, we have so much trouble culture it. If we culture things in monoculture, sometimes they're just so they're happy to have nutrients and they're happy to be all by themselves and they don't produce chemistry.
Yes.
so so we spend a lot of time trying to sort of make things produce chemistry in the lab, and we've gotten pretty good at it in different ways, both with special types of co-culture, studying organisms in situ from the field, but also these types of co-culture using elicitors to make things produce in the lab also. And so from the massive suites of data, we can use the ecological insights, the experimental design to whittle down to where we have maybe one of what are called nodes in these suites or haystacks of data, to where we can say, okay, we know that that molecule is lincomycin. And so in the slide that we're showing here, lincomycin was one of the really cool things that we found from our squid egg. Well, it turns out that lincomycin is also an FDA approved antibiotic. And so how cool that that's one of the things we found in our squid egg. And as much as that story is really an amazing story all by itself, in the same slide, we're quite interested in the unknowns as well. And so things where we might get this really complex NMR spectrum, which is what's shown there. And we get that complex spectrum, and then what? And so that's where the chemistry really starts to get exciting, because now we have unknown molecules that we spend quite a bit of time figuring out what they do.
So a couple of things for listeners and viewers is I'm going to put in the show notes some definitions for some of these analytical techniques. I'm also going to put a link to your quite lovely website that has lots of information on it. Keep in mind that what we're looking at here are all these mysteries. And I have to tell you, I like a question where I don't know the answer. A lot of scientists don't, and I'm not hating on them. I don't drink the haterade on this one. I don't eat hater tots on it. It's really okay. But to me, I get excited when I see something unexpected and I don't know what it does yet, because I want to know more. And this idea of organisms working together, someone might say, for example, well, why would we care about a tunicate having microbes associated with it? Well, sometimes we find biologically active molecules that way. In addition, there's ample evidence that many microbes can actually affect the development of animals. And we don't necessarily know those triggers, and they're certainly due to microbes. What are they? Your approach gives us those kinds of answers. So we've looked a little bit at the squid, and I love the nidamental gland story. I think it's a great one. I love the tunicates, and I think you have more things to talk about if you'd like.
Sure. So the other one that we've been studying for quite some time is the fungus garden ant system. And I have to tell you, I was not one of those kids that thought that ant farms were cool. I just didn't care. I didn't have time. I don't know what I was focusing on at the time, but I have become one of those adults that thinks that ant farms are really amazing. But interestingly, these fungus garden ants are the thing that I'm fascinated by is how similar what they do is to what we do when we are farming.
Yes.
And so Trachomyrmex septentrionalis is they're the ones that we study, the ant that we study in North America with our collaborator Jonathan Clausen. And Jonathan will go and collect, and I actually got to collect with him once and had a lot of fun. Although one of the things I love to tell chemistry audiences is that you actually mouth pipette the ant.
Yes, you do.
Because chemistry audiences will kind of freak out, because mouth pipetting is like, no, don't do that. But in the reality, it's a whole system that's set up in order to collect the ants while you're collecting their garden. because you need both of them to recapitulate what you see in the wild back in the lab. And so what's amazing is the ants will build these really intricate gardens with using fungus. And so they have it's called Leucoagaricus is the genus, and they take this particular fungus and they'll build it into, that's about the size of a softball, these trichomyrmex. Although there are other fungus garden ants in more tropical regions that make these huge gardens. Here in North America, the ones that we study are about the size of a softball with hundreds of ants sort of in and out and around, and they bring from the surface, these gardens are underground, and so they bring the garden, the ant feeds the garden pine needles or leaves, and so one of the more I cannot think of the word. One of the sort of charismatic parts of one of the more charismatic ant species that many of your viewers will know is the fungus garden ant. Not the fungus, the leaf cutter.
That's correct.
So those leaf cutter ants are taking those tiny little leaves and they're taking them down into the ground and feeding them to the garden. And so this garden takes the leaves and uses chemistry. So one of our studies that we've done is showing that at the top of the garden where the ants really start to bring the leaf matter, the chemistry changes as it goes through the garden because the fungus itself is actually metabolizing the leaf. And so the ant can't eat the leaf, but the fungus can metabolize the leaf, uses the molecules that come from the leaf to both feed itself as well as to eventually make these, they're called ganglionial sacs. And so, but they're really just little blips of sugar molecules. And so the ants come and instead of eating the whole garden, they eat actually just those little sacs full of sugar that the fungus is feeding it.
They're farming. So, they're farming.
Yeah, they're totally farming. And our most recent study is essentially showing that they also weed the garden. And so if a pathogen comes, so some sort of other fungal pathogen, somehow those ants know, and it is chemistry. So they sense the chemistry being different, and they come and they weed that part of the garden and they take it away so that the garden doesn't get diseased. And so, and we figured out some of the molecules that it's working towards, that it senses. We just don't know if it's sensing molecules from the fungus or from the pathogen, the beneficial fungus or the food farming fungus, or from the pathogen fungus. And so that's one of the things that we're working on now, is to show that what's happening, where are they getting those molecular cues. Many people have studied what is another part of this system that is also really exciting, and there are actually drugs starting to become, I don't think they're in the market yet, but they are starting clinical trials from the molecules that live on the ant's chest. And they're not molecules that live there, they're actually bacteria. So there's a bacterium called Pseudonocardia that grows and lives on the ant's chest, and that bacteria makes molecules that protect the whole garden also. So they have like their own herbicide essentially that their fungicide, and very selective because it's not killing the fungus of interest, it's killing the pathogen fungus. It's an amazing system, and thus that's sort of wraps back around to me not being the kid who thought that the ant gardens were super awesome, to now being like, oh my gosh, the ant gardens are super awesome.
So I have to say, I'm fairly familiar with the leaf cutters. Actually, when I went to Panama, I got a model, right?
Perfect.
I love leaf cutter ants. And many, many years ago, when I met Cameron Curry, he gave me a tour of his lab, and he was like trying to show me what a queen looked like. And while he's talking to me, very earnest, very Canadian, very nice gentleman, he's picking the soldier ants off of him and throwing them back in to the tank while he's showing, and I'm sitting here like this. And I'm not anti-insect at all. But the thing is, it's such a wonderful teacher. And I believe there's stuff in many cultures suggesting if you look at ants, they're teaching you important lessons. Well, here we've learned that they did farming long before we were ever on the scene. And I love the different elements that you're talking about. And I want to emphasize to people that we tend to be reductionist in our approaches. And there's a very famous microbiologist named Julian Davies that I was happy to meet with. And he was especially interested in small molecules because people didn't study them very much. And so he said, I want my own omics. He wanted to give it a name. And I said, well, parvus is Latin for small, so we'll call it the parvome. So there you go. I don't know if it's caught on, but this idea of looking at the universe of small molecules and the things that they do is interesting. We tend to want things to be simple, but actually, if things were simple, our lives would not be as rich as they are. Look at the kinds of things that you wouldn't expect. You wouldn't expect to find an antibacterial or antifungal agent in a fungus garden that ants run. But you can. You wouldn't expect to see them on a squid, but you do. You wouldn't expect to see them with a marine tunicate, but you can. And I think nature is a wonderful teacher, is what I'm trying to say.
Absolutely. And I think the next transition, both as I've moved to a new university a couple years ago, as well as from this thought process in what you're saying with the ants and the squid and the tunicate, is they're making these molecules that are super interesting. But guess what? We as humans have microbes all over us.
Yes.
And I'm sure that in your 66 other Microbial Matters podcasts, you've talked about those, at least those bacteria, both on our skin, in our guts. So there are other places that humans have bacteria. And so one of the most exciting things that we've been doing more recently is starting to focus on diseases that are well studied because of their importance to humans. So colorectal cancer would be one of them. And but are also well studied because of the microbial relationship. And so we know that human feces have lots of microbes. What we know a lot less of is the chemistry that those microbes make and how they interact. Is there good chemistry so in a healthy patient that doesn't have colorectal cancer? Does that patient have good chemistry from some microbe that's, this is where calling them beneficial will get me in trouble. But so for some commensal microbe, or is there bad chemistry? Is there chemistry that's producing or part of carcinogenesis or cancer development? And so are there microbes that are particularly challenging for different patients? And so other folks have been studying that for a while. We are most interested in the molecules that's being produced, both in good ways or bad ways. I hesitate to say bad, I suppose, because who knows? They could be bad for one person and not for another. The other thing that we're doing with this is mass spectrometry. This instrument that I've talked about a few times is so sensitive that it will find things really far that are only a little bit there. And so then we can start to know, are there biomarkers? Is there something in a patient who doesn't yet have cancer, but develops towards that? Can we find those markers? And we are most interested in the microbial ones because that's our specialty. But to be fair, there are molecules from humans that could be biomarkers too. We do that.
You know what this reminds me of a little bit? Well, I wanted to say two things really quickly. Arturo Casadevall and I were talking one time about how we define the term pathogen, because he doesn't like that term. And a student of mine once misspelled it to pathogon, and he really liked that. And what I told Arturo, and I think this is true, that in many ways, I mean, we have beneficial, certainly beneficial bacteria in our gut that make, for example, vitamins that we need. If we have physical damage that releases those bacteria into the peritoneal cavity, we're going to have sepsis and have a very difficult time surviving. So like a real estate agent that says that the key is location, 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 I would argue in the microbial world, it's context, context, context. It's the other microbes that are there. And that's why our focus on one particular microbe at a time is dangerous. I'm not saying it didn't have to be that way. It should. But it's important to remember that that's going on. And I've seen this approach referred to many times, and it reminds me a little bit of the story of there are now dogs that can apparently smell COVID-19, or they can smell certain forms of cancer. So what are they smelling? Volatile chemicals that are being made by the disease state. Now, some of those molecules could be made by microbes that are responding to changes in the body of the afflicted individual. And given the sensitivity of mass spec, this is a wonderful thing.
It is, but don't talk to me about volatiles. So only because they're really challenging to work
Yes.
with. And so the folks that work with the volatile compounds, I have huge respect for. And we have done a little bit, but it is so challenging to work with volatile chemistry. We are much more accustomed to working with things that are in liquid, just because they're so hard. But there's amazing chemistry.
So you know the story of geosmin being made by certain actinobacteria, and it apparently brings very small arthropods to them. It smells just like potting soil. It brings these very small arthropods to them like springtails, and then they're carrying the spores off to new locations. And one of my students said, well, could you get a mutant that doesn't make geosmin? And I'm thinking, if you know how geosmin's made, you could look for genes that would do that. But if you're looking for something that isn't making a volatile, how on earth would you test for that? So I agree with you that volatiles are challenging.
What you just mentioned actually leads us to one of the parts of our types of investigation that helps a lot. And so we look for biosynthetic gene clusters. And so secondary metabolites, or specialized metabolites, again, microbiologists aren't the only people who like to use special words. And so they used to be called secondary metabolites because they were different from primary. So primary metabolites were just things that helped you survive and eat and metabolize the different things that you needed to do when you're either in a body or in a host or whatever. And so the secondary ones were for things like defense. And so it was these defensive secondary metabolites that people got started with. Because we now know that they're not all for defense, the field of natural products has started to call them specialized metabolites. And regardless of what we call them, many of them are well known to be formed by these biosynthetic gene clusters. So we often will take our organisms of interest, and so our bacteria of interest, and get a whole genome sequence. There are folks that are doing this with metagenomes also, and we have started to dip our toes into that, although that's also one of those spaces that is towards the edge of what we've been doing. And so, but the whole genome sequence, you can find sort of almost like cartridges, is what I would think of them as. And really, cartridge is a great way to think of it, because one of the other things that happens is that these cartridges or biosynthetic gene clusters can get passed from organism to organism. And so I'm sure you and your classes and your other speakers have talked about horizontal gene transfer, and a lot of secondary metabolites will do this. And so, but what's fascinating is the molecule that you mentioned that I unfortunately am not familiar with, but actinobacteria are so well studied that the likelihood is that there's a genome sequence for whatever bacteria makes that, and that we can find matches to the way that genome is structured that will teach us that bacteria makes that compound.
Yes.
And so we can do some of that without the volatile chemistry.
No, I think it's a wonderful tale, and you know, it's just we don't think about it that way, and I have to laugh because there's so many aromas, or so many colors, or so many things that we see that are just kind of the reflection of the metabolism that's going on, and that's wonderful to think about. I believe you have a honeybee story for us.
I do, although it is a challenging story, and so the honeybee is well known for to have some sorts of parasites that cause colony collapse. And that colony collapse has been well studied. What was not as well studied until recently is that some honeybee colonies will get overcome by fungus. And why do some get overcome by fungal pathogen and others don't? And so this is where the collaboration got started, is then thinking about what the bacteria was. And so there's a bacterium called Bombella apis that that the Irene's lab has shown that it is antifungal. And so it produces some sort of antifungal metabolite. And the challenging part of the story is that we then got started talking and we developed this quite nice collaboration in which we were looking for the molecule. And so this is the kind of story that where we threw all of the things at this problem because we couldn't get the bacterium to produce the chemistry all by itself. And so, or in quantities that we could measure. And so whatever it was that we were doing wasn't quite right. And so we have a genome for several of the strains. And so we have some idea of what it might be. And so we have done many, many different types of experiments to figure out how to find this molecule. And so one of them is incorporating stable isotope labels. And so we feed the bacteria these labeled sort of precursor molecules. The bacteria takes those up, and then we see where those precursor molecules show up on our mass spec. And so we're hoping we're at the end stages of that feeding experiment. And we do have the mass spec data for it. And so we are hoping against hope that that helps us to identify which molecule is bioactive, because all of the traditional things that we have always done. So one of them is bioassay guided fractionation. So you grow up a whole slew of this type of this culture, and you fractionate it into small pieces, and you test those extracts and fractions against your fungal pathogen of interest. And so we have really almost everything in the kitchen sink is how I would describe our efforts with this. And I think we're towards the end of that story, but it has been an incredibly challenging. It's a really good example for both, what I forgot what you're calling them, the micronauts, the experienced and the novice micronauts, to start sort of think about not all of the, by the time something gets to a manuscript, it's done. And so somebody figured it out. And so that's great. But this is a perfect story of, we have this really cool symbiosis, or at least commensal bacterium that lives with the honeybee. And we know it has some some biological activity. And unfortunately, we just haven't been able to ID that compound yet. So we keep throwing the kitchen sink at it. And I think it will be a really neat story to tell someday in the next year or so.
So, Marcy, a question that a lot of people listening or viewing might have is, how do these symbioses occur? How do they evolve? I mean, is it love at first sight, or is there a series of steps that have to take place? Do you have thoughts on that?
I do. I think that there are quite a few different research groups who have studied the evolution of these, especially the squid and the Vibrio fischeri, as well as the ants, the evolution of the ant symbiosis. I don't know exactly how I'm not as familiar with that research as I should be. I do think that there's probably some love at first sight, or potentially thinking back about the evolution, the immune modulation that I had mentioned earlier. It's possible that the bacteria was just originally looking for a home and some nutrients, and it happened upon this particular host, and it's like, oh, this is great. The other reality is, though, that there are probably other chemical cues. And so one of the things I would love to do, but it's very challenging, is to study how it particularly is interesting there in the Hawaiian bobtail squid accessory nidamental gland, because I didn't mention this earlier, but those the ANG is filled with tubules. Those tubules are actually open when there are no bacteria there, and they are open when the squid develops the organ. And so how does that happen? So it gets to be an interesting question. Is it love at first sight? Does a bacteria come into that tubule and then it closes and that's it? Like that bacterial cue? But the reality is, we know that the ANG is not the same, the bacterial population, as the seawater. So there's got to be some selection.
Absolutely.
And so there's selection on all sorts of levels. And so one of the questions I still have and don't entirely know how to answer is, are there things on a beneficial microbe or commensal microbe? Are there parts of the chemistry that produces that triggers instead of just the first one, it kicks out the first one because it's not very interested in it. But along comes another one, and that particular bacterium has a trigger molecule that it produces that allows it to close. I have no idea. This is still a very open question, but it is an interesting story. I do know that the ant story has a ton of people that have looked at the
Mhm.
evolution of that symbiosis, and they know that it's millions of years
Oh,
old and
yeah.
that they have continuously evolved. And in fact, they evolve with their pathogens as well.
This is your podcast, not mine, but there's something that we should talk about sometime called X bacteria. It's not a Saturday Night Live skit. And it suggests that there are some situations that symbioses can occur very quickly. So my belief is some taxa are preset to form associations, but I can't prove that. So another time I'll talk to you about that. Related to that, can I ask a question? And I think it's an interesting one. When you talk about some of the chemicals that are being made by a symbiont, and you mentioned sometimes that you can't find them when you're trying to test things out. Is it possible that there are interactions between organisms, an emergent property, where you get the product you're looking for with these organisms working together that would be kind of hard to do in the lab?
I think so. I think that there are, the honeybee is a great example of a challenging spot where we don't know what the chemistry, we're not sure what it is, and it doesn't seem to want to produce it when we try. And so, are there ways in which these molecules are induced? And I think that there are. There are potentially chemical cues. There are things like the type 6 secretion systems and other secretion systems where the bacteria must be touching. So, two different bacteria
Yes.
must be touching. We often think of them injecting toxin, right? And one bacteria is toxic to another. But the reality is that they could be an inducing factor. And
Mhm.
so we just don't know. And so there's lots of really exciting questions for science out there.
I agree. And isn't it funny that the way we look at a question can sometimes affect what we see? We think we're being open to all things, and we are not. Interesting.
Right.
Do you do you believe that there is a common language of symbiosis? That is to say, are there certain molecules that are always being found?
So, this is a great question. It is part of what I like to tell grant reviewers when I'm telling them about how exciting the work is that we do. Um, and interestingly, I think that my research group is uniquely poised to answer part of that question because we have all these different organisms ranging from honeybee to tunicate to squid to ant to fungus to bacteria to human, like all of these different systems. And it should be that there's some language of symbiosis. I do think that there are specialized parts of that. And so I think that the ant probably communicates differently with its pathogenic fungus than it does with its fungus food. And so I think that that there is potentially an underlying language of symbiosis. I also think there are specialized parts. So it's not a very fulfilling answer because I do think it's sort of both sides of the coin.
Students get really irritable with me when they'll ask me, do I think it's A or B in microbiology? And I'll say yes. Well, like the three different models of how viruses evolved. Which one do you think works, Doc? And I'll say, yes. And
Right.
I'm not wrong. And sometimes things are that complicated, but I love your underlying melody of the symbiosis, and then you can have things built on top of it. And I wonder about that because of how the fact that mitochondria and chloroplasts were originally free-living bacteria that were co-opted by early eukaryotic cells. So there might be a common language. A question about the fungus gardening, and I was struck by the behavior, especially the weeding behavior. And then the question becomes, how can that evolve? And I have to admit that I tend to look at insects as like little robots that have switches. And that's easy for me. And maybe we're just bigger with more switches. But how do you think a behavior like that, because it clearly happened in response to this specific association, do you have any ideas how that might have occurred?
If I had to guess, and again, this would be my educated guess, I suppose. If I had to guess, that is because the ants can't digest the leaves
Mm,
or pine needles
yeah.
themselves. So the detritus, the leaves, the pine needles, the ants can't do that. So for whatever reason, whether that this would be an interesting question to sort of explore the chicken and the egg, what happened first. But presumably, in this particular association, at some point, the ant evolved to not be able to digest its own sort of
Mhm.
the thing that is the food source that is most close to it. And so I think over time, how did it find the fungus to do that? This is a great question. Maybe the fungus didn't used to be underground.
Mhm.
Maybe the ants, or maybe the ants used to burrow down and leave the leaves and get frustrated because they couldn't eat them. And then all of a sudden the fungus, I mean, the myriad ways that this could have possibly happened they're really intriguing questions, although I think it would be really hard to scientifically answer
Oh,
them.
I agree. And one thing to keep in mind is we know that parasites can alter behavior of organisms in quite specific ways to make them crawl up rather than down, that kind of thing. Again, coming back to switches. And so that has to be part of the gardening behavior that you don't normally see in an ant. That those so switches got pulled, which is an interesting
It's hard to
thought.
say. Something
Yeah.
happened.
Yeah. No, I also like reminding people that we don't have the answers to everything. It's really common for someone to kind of pick up a particular technology and kind of hit you over the head with it and saying, well, this is how we find the answer. That isn't always true. Um, Gently put, microbes have their own agenda and it's not ours. And, we want to bend them to our will and we can't. I have an expression, first evolved, last extinct. That's the microbial world. And it really is true. So I have a lot of respect for it. I was once accused of being kind of like spiritual about microbes. Well, that's a separate issue. And I'm not making that claim. What I am saying is there's a lot of sophistication that we tend to not associate with that life because we think of it as simple. And it isn't. It's vast and various, to borrow from Stephen Jay Gould and from Charles Darwin. And it really is true, that what you're looking at here, and this is what I love about your work, and I mean this, is the association between organisms, because people tend to be reductionist and they look at one thing at a time. And you have to be so very cautious about it. And it's really interesting because I've had the expression. I mean, Bill Metcalf at University of Illinois is the one who told me this many years ago. I had done a phenotypic assay for something where I would get a red colony on a plate, and that meant something to me. And he said to me, Mark, that assay is yours. The bacteria have their own agenda, right? And in fact, he was right. It wasn't what I thought it was, but it still made a red colony. And bacteria aren't coming from Sigma. They've existed in nature and do all these different things. We tend to focus a little bit on lab rats, things that have undergone domestication for long periods of time. And I don't know that that's the answer either. I'm getting philosophical, but the point is you have all these wonderful interactive systems to look at. You're really driving home the fact that science is collaborative. The ups and the downs, you've talked about why it's important. Even if you didn't think it was necessarily to an outsider, now we've convinced the outsiders it is important. And it's central, I would argue, this ability to interact with our microbial partners. So I have one more thing to ask you, if I may.
Sure.
I like to ask people, how did they become interested or fascinated or starstruck or microbe struck with the microbial sciences?
I thought you were going to ask me how I became a microbiologist.
Well, you can do that if you wish.
I will say that in my brain, I am a chemist.
Yeah.
And I study microbes. And I think the answer comes from sort of the training I had along the way. And so I started, well, I don't want to go into the whole story because it would take us a lot of time. But I think when I first got excited about microbes, um, became it comes from, so my PhD was in plant chemistry. And terrestrial plants make some of the most exciting chemistry
I know.
in the planet.
Yes.
But if you go and collect a plant and you might need a kilogram of plant material, you can't usually go and collect it again, especially if it's from some faraway place. So you're restricted to what you have in your hands. And that has been incredibly successful. The number of drugs we have from terrestrial plants is very high.
Hmm.
So Taxol would be one that many of
Right.
your listeners would have heard of. And it's incredibly useful for lots of different types of cancer. It's a really important molecule, and it's only one of the examples. Um, and so I worked on plant chemistry, and then I worked on cyanobacterial chemistry. And as much as I think of them sometimes as microbes, and I think your listeners will definitely think of them as microbes, we were mostly looking at filamentous
Yeah.
cyanobacteria. The filamentous cyanobacteria, they live in all these filaments. I mean, some of them were like this tall,
Hmm.
like they were
Yeah.
massive, sort of blooms of cyanobacteria. And so we looked at that during my postdoc. And so as I started becoming more of a senior chemist and a senior scientist and sort of understanding what things were happening where, I wanted to find a niche for myself.
Hmm.
And I have always been fascinated by how things interact. I love to go on a walk in the woods and think about, sort of let things go and just start to look around and think about, oh, how come that's happening, or
Hmm.
what's happening over there, or and I don't scientifically I don't get really engaged in that necessarily during that hike in the woods, but I just think of it as part of my personality to wonder about how things are interacting. And so it was a natural sort of shift to then think about, as the field of natural products chemistry started to know more and more about how these macroorganisms
Hmm.
were producing chemistry that actually was probably from their microbes.
Yes.
And so we got started with tunicates. And our next was with squid, and our collaboration that I've already talked about. Then ants, then honeybee, then humans, and now we're into mouse experiments in order to replicate human. And so, how did I become a microbiologist? I don't know. I think 2 years ago when I joined a microbiology department was the first time I started to think of myself as a microbiologist, because I am absolutely, in my brain, I am a chemist and who studies microbial interactions and the chemistry that happens between them.
You know, it's interesting because I'm trained as a microbial geneticist, and I once gave a talk, and a very famous, if somewhat indelicate scientist once called me an ecologist, and I am far from an ecologist. And I said, well, no, I'm actually a microbial, and you actually probably know who I'm talking about. And I said, no, I'm a microbial geneticist. He goes, no, you're not. You're an ecologist. And he was like argumentative with me. And so finally I just let it drop. But it is interesting as time goes on, I'm more and more interested in the interactions between organisms. And that is sort of kind of ecology adjacent, as opposed to changing a nucleotide in a promoter all the time. And there's nothing wrong with either direction. It's just where, the heart wants what the heart wants, as the saying goes. Something that I love to tell people when they look at their skin, and I had an artist friend of mine make this illustration of people holding hands, and there are little dots to represent the microbes, and it's to say when you're holding hands, so are they. And it's really interesting to think about the microbes on your skin, as you mentioned a few minutes ago. Some of them make these short chain fatty acids that prevent other bacteria from growing. So I'm not giving everything kind of a martial, aggressive, militaristic view, but everything's about interaction and real estate. And you're we are all a series of niches when you think about it.
Yeah.
I have to tell you, Marcy, this has been a delightful podcast. I like the way you think. I like your broad interests. I love how you discuss your collaborative work. And I can't wait to have you on the show again to talk about further things that you're doing. And I just want to take this opportunity to wish you and your lab group and family and friends the very best.
Well, thanks so much for having me, Mark. It's been a delight to get to know you better and to talk about our work.
Thank you so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as usual, tasty links indeed, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the Biology Department of the University of Puget Sound in rainy Tacoma, Washington. Dr. Marcy Balunas is Associate Professor of Microbiology and Immunology with a joint appointment in the Department of Medicinal Chemistry at the University of Michigan. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music that makes me smile every time. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.